Mitochondria. Its structure is bounded by a double membrane. The inner membrane has infolding known as cristae. It usually have a range of size of 1 to 10 micrometers long. The function is cellular respiration. The endosymbiont theory is a theory that proposes that the ancestors of the mitochondria were oxygen using non-photosynthetic prokaryotes. Over time, the mitochondria were engulfed by the cell, and they were eventually grew to stay within the cell.
Plants, animals,
that is how
fungi,
scientists
and
believe
protists.
they stay inside
Some cells
them.
have a single large mitochondrion. However, most cells have hundreds or even thousands of mitochondria. This number correlates with the cell's metabolic activity. Mitochondria is enclosed by two membranes, each composed of a phospholipid bilayer with unique collection of embedded proteins. The outer membrane is smooth, and the inner membrane is convoluted with cristae, or foldings. Cristae give mitochondrial membrane a large surface area, which enhances the productivity of cellular respiration in the cell. The inner membrane has two compartments. The first compartment, the inner membrane space, is narrow space between the inner and outer membranes. Compartment 2, the mitochondrial matrix, the area enclosed by the inner membrane that contains many different enzymes, as well as the mitochondrial DNA and ribosomes. The enzymes catalyze some steps of the cellular respiration. The mitochondria has many stages, the first being glycolysis, where glucose is turned into pyruvic acid. Stage 2 is Krebs cycle, where CO2 is pumped out using the electrons carried by NADH. Stage 3 is electron transport change, using ATP synthesis action. In these three stages, ATP is created and pumped out to all parts of the cell. What is the cell wall? The cell wall is the outer layer that maintains the cell's shape and protects cell from mechanical damage. It's made of cellulose, other polysaccharides, and protein. What is the cell wall made of? Cell walls are made of cellulose, which are only found around the plant cells and few other organisms. A cellulose is an organic compound known as a polysaccharide. What is cellulose? Cellulose is a specialized sugar that is classified as a structural carbohydrate and not used for energy. How does the cell wall do this? A great analogy of a plant cell wall is the cell is like a water balloon, and the cell wall is like a cardboard box that protects the balloon. It maintains its shape but can also be penetrated. Is the cell impenetrable? No, it's not. There are small holes called plasmodesmata in the cell walls between plant cells. The cell membrane of neighboring cells are able to connect through these holes. The connections all allow the transfer of nutrients, waste, and ions. The extracellular matrix is a collection of extracellular molecules secreted by cells that provide structural and biochemical support to the surrounding cells. The main ingredient of the extracellular membrane are glycoproteins and other carbohydrate-containing molecules secreted by the cell. Glycoproteins are proteins with covalently bonded carbohydrates, usually with a short chain of sugar. The most abundant glycoprotein in the ECM of most animal cells is a collagens, which forms fibers outside of the cell. In eukaryotic cells, mainly in humans, collagens account for about 40% of the total protein. Cells also attach themselves to the ECM by proteins called fibronectin. Fibronectin bind to the cell surface receptor proteins called integrins that are built to the plasma membrane. Integrins span the membrane and bind on their cytoplasmic side to associate proteins attached to microfilaments of the cytoskeleton. Integrins are in a position to transmit signals between the ECM and the cytoskeleton and thus integrate changes occurring outside and inside the cell. By communicating with a cell through integrins, the ECM can regulate a cell's behavior. The extracellular matrix around a cell can influence the activity of genes in the nucleus. Information about the ECM probably reaches the nucleus by a combination of mechanical and chemical signaling pathways. Mechanical signaling involves fibronetic integrins and microfilaments of the cytoskeleton. Changes in the cytoskeleton may, in turn, trigger chemical signaling pathways inside the cell, leading to the changes
in the set of proteins being made by the cell and therefore changes the cell's functions.